Biochemistry of nucleic acids. Nucleic acid, naturally occurring chemical compound that is capable of being broken down to yield phosphoric acid, sugars, and admixtures of organic bases, purines, and pyrimidines. Nucleic acids are the main information-carrying molecules of the cell, and by directing the process of protein synthesis, they determine the inherited characteristics of every living thing. The two main classes of nucleic acids are deoxyribon nucleic acid or DNA and ribonucleic acid RNA. DNA is the master blueprint for life and constitutes the genetic material in all free living organisms and most viruses. RNA is the genetic material of certain viruses, but it is also found in all living cells, where it can play an important role in certain processes such as the making of proteins. Nucleotides are synthesized from readily available precursors in the cell. The ribose phosphate portion of both purine and pyrimidine nucleotides is synthesized from glucose via the pentose phosphate pathway. The 6-atom pyrimidine ring is synthesized first and subsequently attached to the ribose phosphate. The two rings in purines are synthesized while attached to the ribose phosphate during the assembly of adenine or guanine nucleosides. In both cases, the end product is a nucleotide carrying a phosphate attached to the 5-carbon on the sugar. Finally, a specialized enzyme called kinase adds two phosphate groups using adenosine triphosphate or ATP as the phosphate donor to form a ribonucleoside triphosphate, the immediate precursor of RNA. For DNA, the 2-hydroxyl group is removed from the ribonucleus diphosphate to give deoxyribonucleoside diphosphate. An additional phosphate group from ATP is then added by another kinase to form a deoxyribonucleoside triphosphate, the immediate precursor of DNA. DNA is a polymer of the 4 nucleotides A, C, G, and T, which are joined through a backbone of alternating phosphate and deoxyribose sugar residues. These nitrogen C containing bases occur in complementary pairs as determined by their ability to form hydrogen bonds between them. A always pairs with T through 2 hydrogen bonds, and G always pairs with C through 3 hydrogen bonds. The spans of A, T, and GC hydrogen bonded pairs are nearly identical, allowing them to bridge the sugar phosphate chains uniformly. This structure, along with the molecule's chemical stability, makes DNA the ideal genetic material. The bonding between complementary bases also provides a mechanism for the replication of DNA and the transmission of genetic information. The strands of DNA double helix are held together by hydrogen bonding interaction between complementary base pairs. Heating DNA in solution easily breaks these hydrogen bonds, allowing the two strands to separate, a process called denaturation or melting. The two strands may reassociate when the solution calls or forming the starting DNA duplex A process called renaturation or hybridization.